So next up, we have Dr. Bernd Blossey presenting on updates on the development of biological control for knotweeds, Phragmites, and water chestnut. And I'm pleased to introduce Bernd. Bernd was born and raised in northern Germany. In 1992, he moved to Cornell University, where he is a professor directing the Ecology and Management of Invasive Plants program in the Department of Natural Resources and the Environment. Bernd develops and implements biological weed control programs. Among his target plants are purple loosestrife, water chestnut, Japanese knotweeds, and invasive Phragmites. An ever-increasing focus of his team are investigations into impacts of multiple stressors, including invasive plants, earthworms, slugs, and deer, on a wide range of native organisms. He is intimately involved in different approaches to deer management at Cornell and in the surrounding municipalities. He has developed a network of deer exclosures to study impact of deer on many species and processes, and is developing bioindicators to assess effects of different stressors, including deer. He has also recently completed a large study in northern Wisconsin to assess the effects of wolves on deer and forest plants, a trophic cascade. The ultimate aim of this work is to increase the conservation values of all lands through development of best management practices. Please extend a warm welcome to Dr. Bernd Blossey.
Thank you so much, Abby.
And I will stop
Is
sharing so you
the
can share.
is the sound okay?
Sound is great.
Okay, so let me share my presentation here, and hopefully that all worked out. And you confirm that, Abby?
Yep, I can see it.
Okay,
Perfect.
perfect. So thanks for the invitation. You're catching me halfway between packing and running to the airport to fly over to Germany. So Abby timed this perfectly. Um, I will give a presentation that some of you may have seen before that was at the Invasive Species Expo. The slides will be the same, the words will be different, but conveying some of the same messages that I have. So while the title gives an update on biocontrol of knotweed and Phragmites and water chestnut, I will dive back a little deeper into the history of biological control because there are some very promising aspects and some rather disappointing aspects with it. So bear with me in what I'm trying to tell you here. So here's just a little overview of some of the species that I've worked with. You recognize all of them. You may not recognize the beauty of uh, purple loosestrife here in the center with Tracy standing in there at the Montezuma National Wildlife Refuge. Well, we'll get to that. If you have questions about some of the things, some of the associated papers that I may not have acknowledged here on screen, where you can do a screen grab or so, all the papers that we are publishing right now, with maybe very few exceptions, are publicly available without a paywall. So get in touch with me and I will send them to you, or you can just try to find them yourself. You can download them if you want to dive deeper. So let's see. Let's move on. The very, very disappointing news and reality is that the people that did biological control and were the champions of it here in the Northeast or the Eastern US and Canada, they retire. I'm not that far behind. And the unfortunate news is that they're not being replaced. So whether that's Rob Boucher in Canada or Dick Casagrande in Rhode Island and Judy Huff Goldstein, um, in Delaware, those folks are not being replaced. There are very, very few individuals, including in federal agencies, that can help us to develop further some of these methods that are probably the only really successful long-term methods. It's very unfortunate. It will take some public pressure and maybe even the recognition that we can't deal with the non-native species without biological control that may change some of that. So whatever you can do to help convince universities to continue to invest in these positions, it's a very tough sell, I can tell you that. The wonderful news is that biocontrol is actually everywhere. We just don't realize it because most of the species that are introduced, regardless of what they are are actually kept in check. And mostly that is by biological control. Those could be parasitoids, it could be predators, it could be all kinds of other things. And here's just a collection of species that have declined in their novel ranges. Some of you, this is Long Island, may remember the viburnum leaf beetle. I think it was a threat considered all over the place. It no longer is. It boomed and then it busted. And we really don't have any good idea why it is no longer a problem. It's still there. You go to your viburnum species, most of the shrubs have actually survived. The insect is there, but not in outbreaking densities. And nobody has really looked at that. It's most likely some switching of natural enemies, parasitoids, or something else. And here are some of the favorite species that you could account across the world, whether that's mammals like our red squirrel in Scotland, where the native martens are putting a dent on it, or whatever else it is. We have lots of evidence that populations of non-native species that were invasive are collapsing in the new range. So what is important for us is to facilitate biological control if a uh, um, self-regulating or it's developing for whatever reasons, accidental introduction of natural enemies from the native range or built up switching of natural enemies from in the introduced range, whatever it is. So when we manage species, what we do not want to do is interfere with these processes that are already occurring. And that's a very, very tricky situation. And we'll talk a little bit about that in a moment. So I'm going to go back to the example that brought me actually to North America. And so that was purple loosestrife. I came to North America after doing a PhD on purple loosestrife and the associated insects in Europe. I had the wonderful luxury of traveling between central Finland and southern France to look at all these insects. These are the really big hitters on the left side, from flower feeding insects to leaf feeding insects to root feeding insects. And together, the most obvious ones were always the leaf feeders. Over time, over like a 25 over 25 years, what we have seen is the development where the insects were initially released in 1992. We saw a decline in the abundance of purple loosestrife, then an increase in plant diversity, and then after 15 to 20 years, finally something significant, an increase in native plant diversity. That's what we were after, right? So we've tried other things with birds and others, and never was there enough money available to do this. We did this on a short uh, with very limited budgets and lots of volunteer people, but we had the same quadrats in the same wetlands over 25 years. That allowed us to do these things together with Victoria Nuzzo and Stacy Andrus, who has analyzed most of this. But what you can see here, biocontrol takes a lot of patience, it takes a lot of time, but probably is successful because it takes that slow process of reducing the non-native species abundance, allowing native species to recolonize the space that is vacated by loosestrife. If you go in and you spray or you bulldoze or do any other things, you set back the successional stage to the beginning, and you get a lot of the annual species, and then over time, the ones that are producing the most seed, which is very likely a lot of the non-native species, will recolonize and then dominate the system. So you need to have patience. You need to let this play out, which is very, very difficult to convince people that they need to wait and see what's happening. We need to have assessment protocols that is actually ongoing. So you're not just waiting. I wouldn't just wait for Phragmites, for example, to go away. We have long enough of a history that it doesn't just simply go away by waiting. But here we introduced insects that do that. So they initially, as I said, they're established, they overwinter, they reproduce, and very quickly dispersed. And then you get this decline in stem density, the return of plant diversity. And then we call this early on the suppression of a plant, of the host plant of these highly specialized insects. We call it a biological success. That's not the only thing, but in many places we were lucky to also then see an ecological success, which is the return of native plants and native plant richness. That's what we were after. And the interesting thing some of you may be old enough to remember that there was a big debate about whether purple loosestrife actually is the driver of the decline in wetland plant diversity and in other species. Some said it was hydrology, it was other things. Well, we documented with the work that we were having done over the last 20, 30 years that actually the stem density of purple loosestrife is the main driver. If you have very, very high stem densities of purple loosestrife, the total cover of other species is decreased. But what you also can see in this graph here, we don't need to go down to zero. Oops, biological control doesn't want to go down to zero and should not go down to zero. So because we want to maintain some food for the insects so they can maintain populations, and that's very important. So you go below 20 stems per meter square or something of that sort, most of the species will be happy with that. Loosestrife at that time is just another organism in a wetland system. It's also critically important to not try to get this down to zero. So don't dig up the last loosestrife plants. Let them be. They're a fueling station for the insects that are dispersing, trying to find outbreaks of purple loosestrife. You will never get it down. There's a seed bank there. Please do not try to control the last members of purple loosestrife that you have in the community. Just enjoy them. And so, again, these are just reiterating some of the same things that I said with the overview diagram. And it shows how many times it takes before initially the number of stems are declining or the stem density decreases. And it happened, and we released insects in places where purple loosestrife was a very domineering effect, or it was a just another species within the wetland system. In all of those situations, the insects further suppressed purple loosestrife. But as I say here, the return of native plant diversity, it really only became significant in our results after 15 years. So, as I said, patient required pays off, biocontrol successful because it's slow. And the native species can respond. It can actually produce seeds. The seeds may not automatically be available. In wetlands, that may be slightly a different situation because seeds may hang in there for a very, very long time. In forests, we don't have a seed bank that can respond to an ecological opportunity. And so here are just some pictures from what you saw before. You'll see a purple loosestrife in there, but with lots of other native plants that are dominating in there. Sometimes you need to really look for a loosestrife to find it. Sometimes there are years where it's more abundant. It's still there. It's not going to go away, and that is just fine. So one of the papers that we have published called From Ecological Menace to Roadside Attraction. What you will see driving through New York and other places, because this is repeated across the country, you will have lots of roadside populations that are flourishing. And it's in part because roadsides are not the best habitat for the insects. They're being mowed, they're being sprayed, there's salt influence, and that just sets the biocontrol back. So you see loosestrife along the roadsides, who cares, right? So the insects will do their things, and the roadside populations is not what we're worried about. So we'll see that all the time. I'm switching to another one that I have looked at for quite some time, which is garlic mustard. There's a longer story about the garlic mustard. If you want to indulge yourself, you can listen to that on YouTube here on the NISRI YouTube channel. I gave a long presentation after we finally published the results that we had. And so the question here is, remember that I said we need to decide if we intervene, whether we actually set the biological control back or whether we're enhancing it or we're doing something that is useful or actually counterproductive. So I use these slides and say, who's the better manager? And I will tell you, age and wisdom, having a person sitting on a bench is much more successful in terms of garlic mustard control than using and coming and digging up in a sea of garlic mustard. Or you will all have heard and seen the pieces about the garlic mustard challenges, ripping out garlic mustard by the by the bag load. You may have some fun doing that. It is counterproductive and it is not recommended to do that. You are setting the biocontrol back. And why is that? So we did a very long-term study trying to repeat what we did with purple loosestrife, but being prepared for the potential introduction of biocontrol organisms, insects that were studied at that time in Switzerland. And so these are the different locations that we were looking at. across the Midwest and Northeast. And we put all these data together. And then we were trying to figure out what was going on over time. They all started in pretty heavily invaded areas, kind of like what the picture you see on the top left corner. And then we had um, measured the abundance, counted stems, counted height, and cover and other things. But what we realized over time, species or places where garlic mustard was here with the little dinner plate sized leaves and very tall plants, 3, 4 feet tall, over time declined to these little puny things, which are just a few inches high. Not every one is like that, but we have seen that ourselves. The places where we did work, I did that here on our own property, weekend after weekend in the spring, trying to do something about the garlic mustard that we had, only to then see in the places where we didn't do anything, success took care of it. We didn't need to interfere. So what's happening? So here's some of the data. And it takes a long time, once again, and there are some regional differences. So we had between 10 and 30 quadrats at each of those locations. And here's a division between the Midwest, which is here on top, and the Northeast, and how these quadrats were developing by longitude, if you want to call it that way. And you can kind of see that the decline is stronger and faster in the further east we go. That is a reflection of garlic mustard distribution history or range expansions. Initially, as far as we know, it occurred on Long Island, so it comes home to LIISMA, and then it spread west. Went into Chicago some 30 years later or so. We don't know how it all got, but over time, you can see that even in some quadrats, garlic mustard completely disappears. So these are half-meter square quadrats. That doesn't mean that garlic mustard disappears from a site, and it shouldn't disappear from a site, but it happens over time. Here it's a decadal time frame, but it happens faster in the east, which is associated with residence time, how long garlic mustard has been in the region. And if we put all these things together, this is what it's a population growth rate that gets you to a point where how fast do populations grow or decline. When lambda here is at 1, populations are stable. When lambda is above 1, populations are increasing. When it's below 1, populations are declining. So you can see that at each of these places, you need to wait at least a decade or so, or maybe even a little longer, and garlic mustard populations will decline. Garlic mustard will stay in the system. There will be some plants that are nice and tall, maybe a couple of feet high, but the density over time will decline. There's still fluctuations. Sometimes you have a wonderful moist year and garlic mustard comes back from the seed bank. Seed bank can last for 13, 14 years. And so once again, patience pay off. And this is a ripple effect. There are places where garlic mustard has never been. Garlic mustard will colonize those areas, increase and then decrease. Two important things to mention here. Garlic mustard follows worms. You will not find garlic mustard, or maybe there is 1 or 2 locations in North America where garlic mustard occurs where there are no introduced earthworms. Second, the process that I've just described is one of negative soil feedback. So the longer garlic mustard is in the soil, the more microorganisms are being built up that suppress garlic mustard and suppress garlic mustard selectively. So native species are coming back and doing just fine. So it's a very selective biological control. If you go in and you rip out garlic mustard, you mow it, you do other things, that negative soil feedback process will be eliminated and you start not at 0, but you set the entire process back. Please do not weed garlic mustard. The only occasion where I can see that that would be useful if you find a couple of individuals where they have not set seed and the next population is miles and miles away. That I can tolerate. Everything else, learn to live with it. It's also not a threat. I'm not going to go into that one any further. So, as I said, this is the negative soil feedback. It's a process that may be occurring for many, many different species. Here on the left side is a diagram by Dietz et al. From New Zealand that can see how much the negative suppressive effects of negative soil feedback has occurred. And it's a function of how long the species, these are different species, different plant species in New Zealand, how long they have been there. So if you are there for a couple of hundred years, that suppressive effects of soil microbial community is very, very strong. It becomes weaker the less long your residence time is. And we see that here in North America. This is by Warren, who tabulated. I'm not entirely sure what he didn't collect his the data himself, but looked at published data. And this is in woodlands, I believe, yeah, eastern U.S. forests. And the residence time and the abundance and richness declines over time, once again, as a function of how long these species are there. So sometimes we just need to sit there, watch it carefully, and see where it would be declining. Maybe even have areas where we can study it in some detail, because the important thing is to then distinguish the ones that are serious invaders and where this would not happen from the ones where it would happen. So for the species that I'm talking about now, and I'm focusing on non-native Phragmites, Japanese knotweed, that complex of three different species, or two species and its hybrid, and then water chestnut. Those species I consider strong invaders where there is no evidence that the species would be declining. Here is an overview of the Phragmites biocontrol program, how it has developed over time. It was initiated, well, it's now almost 30 years ago. So in 1998, at that time, people were still debating whether it's native or non-native. Now we know it's both. There's a cryptic invasion, so we have an introduced, for now, considered a subspecies. But there are very distinct differences, and I think I gave a presentation to LIISMA a few years ago how to separate those two. We asked questionnaires of wetland, or we asked wetland managers in 2009, but they were thinking about biocontrol. We've repeated that work. I will show you some data right now. Lots of host specificity testing. It's because there's lots of herbivores in Europe, and by having a subspecies, it becomes rather surgical of what you need to find in terms of the organisms that can be introduced. We were satisfied with what we had and submitted a tag petition. That's a technical advisory group for biological control, and that was submitted simultaneously to US and Canadian authorities. The Canadian authorities thought the data are strong, approved the field release, and first releases occurred in 2019, and then through the COVID year of 2020. I can talk a little bit about what's happening there. TAG in the US, or not TAG, TAG, the group approved the petition for field release, but USDA APHIS did not go along with that and didn't issue a field release permit. What they were asking us to do is come up with additional evidence to see whether non-native Phragmites has any utility, and whether the threat to native Phragmites can be better quantified than what we have done, excuse me, in the tag petition. For a little background, I don't know what I have next here. Yeah, so just a little background on non-native Phragmites. Across the US, it's considered one of the most invasive wetland species that we have, except in the Mississippi River Delta. In the Mississippi River Delta, we have the hope by some people that non-native Phragmites can keep the delta together, because with lack of sediment coming in from the Mississippi River or being dumped way out to keep the shipping canals open, it's the Mississippi River Wildlife Refuge that sits out way in the high salinity areas. Phragmites has colonized those places, and they think they keep the delta together. What's happening there is you have subsistence of the delta, you have sea level rise, you have lack of sedimentation, and you have a wonderful South American rodent, the nutria, eat all the native plants in the Mississippi Delta. So native Phragmites is, or I'm sorry, there is no native Phragmites in Louisiana. Introduced Phragmites is collapsing in the river delta, and there was hope that the native Phragmites, I'm sorry, the introduced Phragmites would keep the delta together, and hence no biocontrol should be allowed for the species because of the Mississippi River Delta situation. We have long debated that. There's publications about that. Um, I think it's high salinity. The only way that we can control non-native Phragmites along the coastlines is by keeping it under tidal influence. That's what's happening in the Mississippi Delta. That's why Phragmites is collapsing. But they basically had a lock on not allowing the release at the present time. That may lessen, but there is still USDA APHIS is looking at the Mississippi River Delta and said, what can we do? Our modeling also shows that these organisms that I have here as a life cycle will not go that far south. That's almost subtropical. They will not go that far because they're temperate insects. So they will stick to the temperate parts. I don't know how far south they will go. They're in North Africa, but they will not go to the subtropics. That's just very clear. So we have two organisms. One is called Arcanara, or now called Lignisa geminipuncta, and Arcanorica, that basically look pretty similar. They're stem-feeding moths. As larvae, they feed in the interior of the stem. Eggs overwinter under leaf sheaths. And this is very important because the females make decisions of where to lay their eggs. On native Phragmites, leaf sheaths fall off. Females avoid laying eggs on native Phragmites because the oviposition period is in July and August when those leaf sheaths become loose and drop off the plant. So we have not a chemical but a morphological character that prevents these organisms from laying eggs on native Phragmites. If we force the larvae to just live on a native Phragmites, they will be able to develop on that. So eggs overwinter, hatch when the shoots are about asparagus size. Larvae come in, kill the growing point, mine to 2, 3, 4 different stems, pupate in July, and then there's a flight period in July and August, depending on what it is. The damage is depending on when the larvae are actually active. The lower asparagus type shoots will be killed, so they're not doing any photosynthesis. The higher ones are killed over time. And this is kind of like a depiction, a schematic diagram of what's happening. So here, they change shoots 3 different times. So over the life cycle of a Lignisa or Arcanara larva, 4 different stems will be attacked. The last one, the 5th one, where the pupation occurs, which is in the lower area, will not do any damage. The larvae will go in, make a little window, so the adult come back out. But these 3 or these 4 different ones, shoots that are attacked, will be damaged quite substantially. The evidence from Canada shows that stem densities decline where they have been released. These insects established everywhere where they release them. They have wonderful release procedures. They will come to New York where we're monitoring that, whether they actually jumped across Lake Ontario or the St. Lawrence. It seems that native plant species are doing better in the areas where we have lower shoot densities. I've told you the stems become thin, and native plants are increasing in there. So everything that we would desire from a biocontrol organisms seems to be materializing as early evidence that we have from Canada. So we're following this up. Some of the evidence that we have when larvae go in and kill the growing point, you get secondary shoots. So this is what you see. So Jess McKenney here is holding this up. We're in Aurora in Canada, where these organisms have been released and are heavily attacking that. The one thing that I need to warn you about, native Phragmites does this too when it's being attacked and the growing point is killed. So this is a picture, not from New York, but this is an old volcano in Colorado where there are no invasive Phragmites as far as we know now. It's all native Phragmites. And here are native insects attacking native Phragmites, and you can get these secondary sprouts. So just because you have a secondary or side shoots doesn't mean that it's a biocontrol organism that has attacked us. But so we'll follow this up. We will have monitoring protocols for that. And hopefully we can confirm the presence in New York pretty shortly, because then we would be able to distribute them further. We're testing for the safety of native Phragmites by doing various things in Canada. We can do that there now in much better ways. We will do roadside surveys where we have alternating populations of native and introduced Phragmites. These moths spread about 1 to 3 kilometers per year. They're flying as females, so larvae obviously can't crawl that far. So we have planted in various places native Phragmites in adjacent to introduced Phragmites. As I said, we do roadside surveys, and we will follow up this year whether native Phragmites is being attacked by these flourishing biocontrol organisms from Europe. And we repeated, because APHIS asked us to do that, the attitudes, but we were not asking wetland managers, but we are asking wetland managers in state and federal agencies of what the attitudes are about biological control. And so we have about 130 or so people that responded to our survey. And here are some of the results. They're very similar to what we had earlier. We use a different format in terms of the questionnaire. So let me introduce this graph here to everything to the right side. The darker green it becomes, the more strongly people agree with what we were asking them to do. The more you are on the left side here into the goldenrod color, the less people would agree with that. So here are two questions. So these are not really alternative questions, but people could answer both of them. And consider use of approved herbicides to be an appropriate management method for introduced Phragmites. And you can see 90% of people will agree with that statement. Only 2% would disagree with that statement. And then when it comes to biological control, once again, the overwhelming majority of people would consider it a safe use of biocontrol. And only few of the individuals or a small proportion would consider it not a safe method. I don't know what I have to. Oops. Sorry about that. So then we asked them whether their support for biological control is dependent on how much threat there is to native Phragmites. And you can kind of see if this is hypothetical, this is not realized, right? So if the insects would cause population declines of both introduced and native Phragmites, the support for biological control drops very to very, very low levels. So here, the almost half of the individuals would say we shouldn't be doing biocontrol if there are population declines of native species. On the other hand, you also see that 70% of people would say, do biocontrol if we are unable to stop the spread of introduced Phragmites. So there is a gradient in terms of the support for biological control that depends on how much of a threat biocontrol agents are to native species. If they would never attack native Phragmites, 83% of people would do biocontrol. Remember that we don't have biocontrol champions that are growing up around us right now. So the people that are doing this, the people in Canada or faculty members in the US have retired. So this strong support that we see, if you were to go and target other species that we may want to target that are not Phragmites, it's going to be difficult to find people that can support that kind of work. Go into water chestnut. Many of you will know what it does. I don't know how widespread it is on Long Island, but what we see here I think this is the Mohawk River or the Red River in central New York, with Wade trying to push a canoe through that. If you go to China, you have these situations. So we have a leaf beetle very similar to the ones on loosestrife in the same genus, the Gallericella, called Gallericella brahmanica, doing this damage to Trapa in its native range. Those become never as dense as what we see here on the left side. The larvae the insect can have multiple generations depending on how far north or south it is in China, and those mats will sink. So we've done some modeling and some other things. We think it's a very, very wonderful organism to have here. And so Wade Simmons, who has just completed his PhD in December, and he's staying on with us as a postdoc to complete the water chestnut project here. He's done a whole bunch of work on specificity, impact, and potential hybridization because this organism is very closely related to the native water lily leaf beetle. And we're working on the tag petition right now to put this all together, and then we will see how fast federal agencies are working these days. I have no prediction of what will happen. I'm not going to show you any of the results here because I'm going to jump to Japanese knotweed, but there is very, very high hope for water chestnut to be under control by the water lily leaf. I'm sorry, not by the water lily leaf beetle. It attacks them, but the native one doesn't do any control efforts, but by Galaricella bermanica. So you will have Japanese knotweed all over the place here in its full beauty of its bloom. There was actually an organism, or is an organism, that is being released to control this after some very substantial host specificity testing that was done initially in the UK, because that's a problem in Europe also, and then by Fritzi Gresta in Oregon. And the critter that was released was Aphalara itadori. It's a psyllid, so similar to an aphid. And unfortunately, it's a dud. It's a sucker, but it sucks as a biocontrol agent. So wherever they were releasing it, there are different strains from different regions collected in Japan. And basically, it failed to establish left and right, and whether that was Canada or whether it was in the UK or in the US. And so there are very, very few individuals, and I think there's one population in Oregon where insects have survived, but never built up huge numbers. You can rear them just fine on all the different Phragmites species and the hybrids if you protect them from predators. So here, biological control of this of the psyllid is probably done by lots of predators. Unfortunately, this is biological control of an organism that we would like to be invasive to control another invader. So if you leave them outside and without protection, they just disappear. So what's left, there's some people have some hope that there are some secret populations that are being built up somewhere. I have no hope for that. Or people are experimenting with a different strain called the Murakami strain, which is I don't know whether it will have any influence or not. We'll see. They're trying to petition for the release of the Murakami strain right now in the US. And I think it has been released already in Europe. And it's not doing much because the damage to the individual plants is just not that great. So where we have been working on, and we're waiting for funding and the things that I have seen, I have some funding in place from the DEC, and there's funding that seems to be in development through the New York Department of Transportation, but it hasn't materialized. So there are other organisms that the folks in the UK initially rejected in Japan. There are other leaf beetles here. There's a stem mining weevil. There's a sawfly, which is gregarious. And there's a swift moth here that attacks the root system, which is what we really would like to have. There are questions about the biology of all these organisms because they haven't been well studied. There may be lots of sibling species in among them. So we need to work with Japanese collaborators to figure out, are these safe organisms to be introduced? But that would be the work that I think needs to happen in the next few years, as soon as we can get funding to then work with our Japanese collaborators on that. Um, a couple of words on some of the things that did not work. So swallowwort, also something that you worry about on Long Island. They introduced a moth, Hypena. It's a dapple light specialist. It establishes but never does anything to swallowwort. Uh, Thousands of them have been released. I think Dan Perry or Dylan Perry at SUNY ES have said they may have released 100,000 or so. There's no established in New York of this moth, and there is no impact on it. So Dylan and colleagues at USDA have worked with a leaf beetle, which is a root feeder, and they're putting a petition together right now to ask TAG for a permit to introduce them. Some of the tricky things were native milkweeds, because that's the organism that's the taxon that swallowwort is very closely related to. So they did a lot of testing in southern France to make sure that that leaf beetle is actually a safe one to be introduced. And that's what I have for now. And I don't know why my screen went black, but that's what it is. I'm happy to entertain questions that you have. We have some time. I need to be done by noon because I may get a phone call. So thank you very much. How do you want to deal with this, Abby?
Oh, thank you so much, Bern. We have, I think I saw two clarifying questions about the Phragmites biocontrol, but thank you so much for that
Yeah,
presentation. We'll definitely
I,
get you
I
out
leave
of
the
here.
presentation up in case somebody asks me to go back to a slide or something like that.
That sounds good. So one question was from Jane Fusillo. She said, do the moths control the native and introduced Phragmites or just the introduced variety?
So all the evidence that we have right now is that they have a very high preference for the introduced one. And that's related to the oviposition of the females that like the leaf sheets to be tight on the introduced ones. If the leaf sheets are not tight and stick over the winter, the moths refuse to lay eggs there. They just leave the Phragmites patches. And so what we have seen in Canada so far seems to suggest that are not being found in native Phragmites, but we need to do something more extensive on that one to fulfill regulatory requirements or a request from USDA. There's no evidence to suggest at this point, but it's early in the program that they're attacking native Phragmites. So you need to wait for additional evidence. And I wouldn't say that the moths at this point control non-native Phragmites. They're building up large populations where they can go and they collect stems and bring them to the next site, and they almost have 100% establishment, which is incredible for when you think look, think about just the example that I brought, Hypena, right?
Mm.
100,000 released, no established in New York. They put 100 eggs out there somewhere and they get establishment. What it ultimately then results in the decline so substantial that we can be happy about how wetlands are developing. Please wait. So I can't give a guarantee for that one as of yet.
It's great that it seems promising so far.
It is very promising. So
Yeah.
the Canadians obviously have been involved with Hypena as well. They've done much more work with swallowwort there than what we did here. And
Mhm.
it's just, they're just all over themselves that it works this well.
Yeah. This is a clarifying question on the same topic, that the moth is spreading from Phragmites in Canada into the United States from that introduction in Canada, but it is
No, not yet.
not. But the
Not
person
yet.
was asking if but no biocontrol introductions in the US yet for Phragmites, right?
Correct.
Right.
Correct. So what we are hoping for, and the Canadians, obviously, because we did all the work, and then the Canadians jumped onto the bandwagon and said, "Oh, great that you did all the work on Phragmites. Why don't we just, give you a little bit of money and then we're releasing them here as well?" And now they have funded all this work, so they're paying back in terms of what they're doing. But
Mm.
they're purposefully released close to the US border in the hopes that the insects would travel across. That
Mhm.
has happened for some other programs because the Canadians are so much more open to doing biocontrol and the regulatory minefield that we have in the US for whatever reason. So we have not documented either of these insects that have been released, so both of them are establishing well, by the way, right? So there's two species. We have not documented them in the US as of last year. We haven't looked this year, but we will in June, and that will be the determining factor. Once they're here in New York, we can work with them in New York. And we could then redistribute them in New York because they're in the state.
Mm.
That would be a major difference, right? So we could facilitate the dispersal instead of waiting on the natural dispersal that the moths have.
Okay. Well, that would be exciting. Okay, one other question, I guess related to that knotweed. Do you have any recommended treatments for Japanese knotweed, given that Alpha Laura ididori is not too promising right now? Do you have other treatment recommendations?
Well, my recommendation really is a traditional one. What seems to have worked, um, and it's a lot of effort, is stem injection. And so I don't know whether you have used that in Long Island. I'm
Mhm.
assuming you have treated some places, right? So that's the only one, the one thing that I'm not entirely sure about. is the long-term effects. Once I've seen things in Cornell Botanic Gardens where they went in and treated Japanese knotweed in a forest environment, and they got rid of the Japanese knotweed there, but nothing else ever came back. Now that may be because there are too many deer, so that you don't have native plant species come back, or there may not be a seed bank, so you need to do active restoration. What I do not know is if you do this herbicide treatment, particularly if you were to use imazapyr, that has such a long lifespan, and when they use it for Phragmites, it's terrible. But that's the only thing that I have, or constant mowing and clipping. Uh, you just try to keep it at bay.
Yeah.
That's all. And I'm sure you have the same experiences in Long Island. I'm struggling with two populations here on our own land, and the
Mhm.
tractor is helpful, but last year I went to herbicide, so that was I finally gave up after 20 years. Well, these were small populations, right? And even then I couldn't.
Yeah,
So
you do your own personal trials to test it out.
that's
And
right.
that's what we recommend too, this stem injection if possible. So,
Yeah. Yeah.
okay, and one more. What do you think the method of Acarnara dispersal would be for to control large areas of Phragmites? I'm not sure exactly what is meant there, but maybe you
Right.
can.
So the way that the Canadians are doing it, so there's two methods of dispersal, right? So
Hmm.
one is Acarnara or Lanisia by itself. That means the moths come out, they mate kind of where they are, and then the females go on the wing and they fly. They're
Hmm.
known from Europe as very, very strong flyers. So when you think these are moths, right, there's all kinds of people that try to trap insects at night with black light traps. This seems to be a favorite activity of people that are inclined to study insects or entomologists or so. In Europe, dispersal distances have been documented of hundreds of kilometers. Just an insect flying somewhere. I can't say that it didn't go out, jump on a ship, sat on a ship, and then went off. But where the closest Phragmites populations were hundreds of kilometers away. Here, what the Canadians have documented is dispersal by Acarnara itself, flying distance of 1 to 3 kilometers per year. Okay, so they have found Acarnara that far away from the known release site without them having done anything in addition. We'll go in this spring, I'm sorry, this early summer in June, and try to come up with something more substantial, fundamental, the way that we're trying to follow this along roadsides where we can access that a little better. We'll see where we end up. So the other dispersal mechanism, and that's a human-aided dispersal mechanism. Initially, Canadians were having glued eggs that they were putting out in little cups in different stands to trying to see whether they're getting establishment. So these early trials, some adult releases, so they did that. What they're doing now is in places where the insects have built up nice populations, they go in the fall or in the spring before the larvae would hatch, cut 100 stems, bundle them, and bring them to a new locations, put these stems out, and that is almost 100% establishment. So there you can bring them long distances, of course, that goes beyond the dispersal distance of the insects. So two different factors there.
Hmm. Cool, very interesting. All right, any final questions here? That's all I'm seeing in the chat right now. If not, thank you so much again for joining us, Bernd. It was really informative. I think we all learned a lot, and those are great updates. So
I was
I
a
hope
little
you have.
afraid that you guys all got buried. You probably got buried, but
Oh yeah, no, we're buried. Yeah, there's like 2 feet of snow.
so I'm flying out of Kennedy. I hope they get the runways clear, but everything so far is fine. So thank you very much for the
All right.
opportunity. Enjoy the rest of your symposium, and
Yeah.
I'll see you around.
Have safe travels, Bernd. Thank you.
Thank you. Thank you. Take care.
Okay.
Let me see how I can stop sharing. Oh, I'm
Uh,
there. Okay, I got it.
you got it.
Thank
Great.
you. Bye-bye.
Bye. So, I believe we might take a quick break, maybe a 5-minute break, and then get ready for our next presentation. So take a moment to stretch, and we'll be right back. One last plug for the Resilient Long Island Symposium coming up on March 19th to the 21st. Please sign up. Please join us there. We'd love to see you there, and we'll have more great presentations just like the ones you heard today, in person at Hofstra University and other locations. And lastly, thank you. Our next partners meeting will be hybrid again and on May 15th. Location to be announced, but probably at the Sisters of St. Joseph in Brentwood. And thank you so much for joining us today.